I don't know who was doing that. So we're on.
Hey, Kathleen.
Hey, now.
What are we on? Page 276. Could you pull the blinds open back there? Yeah.
Got you, bro.
Pretty. All right. This diagram kind of shows the eye. If you cut it in half, and we're looking into it, in some of the parts of the eye, there are muscles that can move the eye around, and glands that produce tears. Here are your tear glands. They're called lacrimal glands, and they're kind of located on the outside of the eyes, and they just constantly squirt fluid that goes over the surface of the eye and keeps it moist. See, those cells of the eye, even of the cornea, they're living cells, and all living cells must constantly be in water. So you have to, these glands squirt water out, it kind of goes over the eye. There's an enzyme in that fluid that kills bacteria. So bacteria floating around in the air that land on your eye are dealt with. And then the tears collect and are taken into these glands and actually into your nose, where they help keep your nose moist as well. And so that's why when you see somebody crying, or after I hand back my test, I usually see some kids crying. So the tears well up, and sometimes they tear so much that it just runs down the face. But a lot of it goes down here and runs down your nose and out your nose. So when you see a baby crying or whatever, and it's got stuff running out of its nose, those are tears. It's not snot. Isn't that interesting?
It is.
Probably didn't know that. And so, but normally it's not enough to well up and run out of your nose. It just kind of keeps the eyes lubricated, keeps the nose lubricated. So here's an eyeball, and there are all kinds of muscles of the eye. They've actually cut this one so that you can see in there, can see behind it and such. But there's one that's attached all the way here that pulls the eye toward the outside, and that's the lateral rectus. And there's one that pulls the eye towards the inside, is the medial rectus. And then you have muscles that pull the eye downward, inferior rectus. Muscles that pull the eye upward, superior rectus. And you have a couple muscles that pull the eye. The eye can actually move kind of diagonally, can kind of twist a little bit. And that's the inferior oblique and the superior oblique, which is kind of cool. The superior oblique goes through a little pulley. There's a little pulley right there to turn the muscle this way and pull it so your muscle rotates like that. So it's pretty cool how many different ways the eyes can move. It takes 6 muscles to do it. Isn't that neat? When you're pointing these muscles out to me in the IDs, you have to point the muscles out on the actual model. The muscles aren't on the diagram. Okay? So use the model to show me the muscles. I have an eye model in there that you can take apart and see a lot of this. Of course, kids have damaged it and they stole the lens and all that. So anyway, here's what the inside of the eye looks like. By the way, we're doing an eye dissection tomorrow.
Yes.
A sheep eye we're going to cut up and take a look. It doesn't take too long. It's just a little eye, so it's not as involved as the brain dissection. But anyway, the cornea, that's the clear part of the eye in the front that protects and it allows light to pass, and it bends the light slightly as the light comes through the cornea. And then there's an open space underneath it called the Anterior Cavity, which this basically is everything that's located between the cornea and the lens. We call that the anterior cavity. Anterior means front. And there's a fluid here in this area. It's a kind of viscous fluid. It's kind of thick. It's kind of like a mucousy fluid that fills this whole area. It's called aqueous humor. Humor actually means fluid. If you take the Latin root of it. I don't know how that led to the word humor being funny, but humor means fluid. So we call this aqueous humor that's all filling this area. And you'll see when you cut the sheep eye open, you can see this gooey substance that comes out of the eye. Then there's this part right here. This is called the iris. It's the colored part of your eye. The iris is a muscle that can close like that. When the iris contracts, it kind of closes up. And when it relaxes, it kind of opens this way. It's kind of like a sphincter muscle. But anyway, the hole in the middle of the iris is called the pupil. And so when your eyes dilate, the pupil is large. When the iris constricts, the pupil is small. And that changes the amount of light that gets into the eye. Isn't that cool? Do me a favor, pull those blinds closed for a second. I want you to see how your iris works. You need to find someone near you and look into their eyes. Find someone near you and look into their eyes. Just doesn't hurt. Look into somebody's eyes. You got to get kind of close. And when I turn the light on here, you'll see the iris move. Ready? Eyes open. Look at the iris, the colored part of the person's eye. 1, 2, 3. See the iris move?
Yeah.
Try it again. Try it again. One more time. Look at the iris, the colored part of the eye. When I turn the light on, it should move. It should actually constrict. 1, 2, 3.
We didn't do it that time.
You see it?
No, it didn't.
Okay, pull the blinds back over.
Your eyes are smart. Oh, slow.
The iris, listen up. The iris actually constricts when you see something you don't like. And the iris will dilate when you see something you like. So I always use this to my advantage when I'm in bars looking at girls. Their eyes, if their eyes are dilated, that means they like me. And so I keep talking. And if they're constricted, that means they don't want to see me, so I go away.
But I guess they're always dilated, huh, bro?
It seems that way, yeah. I've noticed the correlation.
Pulling off my...
Now, the lens that's behind the pupil and iris, that focuses the light. The light hits the lens, focuses it on the back of the eye, and the back of the eye we call the retina. This yellow area here is called the retina. And the retina has sensors called rods and cones. Now there's this big space right here called the posterior cavity. And it's filled with a liquid called vitreous humor. That's a little bit different than the aqueous humor that's up here. So humor in the anterior cavity, vitreous humor in the posterior cavity. Are you going to get those mixed up, Michael?
No.
You don't think so?
I don't think so.
I might see a correlation between you not paying attention and getting mixed up.
I was paying attention.
Okay.
What are you talking about?
I was wrong then. The retina on the back here has the cells that sense the light. They're called rods and cones. Rods can sense lightness and darkness and movement, and cones can sense color. So those cells are always sending signals. Each rod and cone has a synapse that eventually sends a signal to the brain, so the brain gets constantly, just right now, just while you're watching, these cells are sending signals to the brain. There's 30 million of them. So you got 30 million signals going to the brain 20 times a second is about how often they fire. That's a lot of energy. The eyes use a ton of energy. A lot of the food you eat goes to running your eyes. So there is a layer right here, this red layer, it's very thin. It's called the choroid coat. That's a layer of blood vessels that surround the retina, and so these cells of the retina, they get nutrients from the blood that's in the choroid coat right here. You also have blood vessels that come out here and spread over the surface of the eye, and those blood vessels also deliver nutrients. And then the white of the eye on the outside is called the sclera. You ever heard the statement, don't fire till you see the whites of their eyes?
Sure do.
Should have been, don't fire till you see the sclera. That would have been shorter.
Wait.
I think it was in the Civil War. They didn't know what it was called.
Sclera.
You can see here the lens is held in place by a bunch of suspensory ligaments, which are these little things that look like wire or lines, and then these ciliary bodies, which are muscles, and those muscles can stretch the lens or relax it. And when you stretch the lens tight, you make the lens thinner, and you're able to focus on objects that are far away. And when you relax, the lens gets thicker, and you're focused on objects that are nearer. So you can control the focus of what you're looking at by stretching the lens or relaxing it. So that's kind of interesting. Here's an eye video.
Our eyes are very mobile with the help of these three sets of muscles. Each pair moves the eyes in a different direction, up, down, or sideways. There's even a little pulley so that a muscle can pull the eye to the side. Inside the eye, there's a succession of components to help us analyze the light and the information it carries. The light enters from the left and passes through the cornea and then the lens. This helps to focus the light on a layer of cells that lines the inside of the eyeball. This is the retina, the orange-colored layer covered with a network of blood vessels. The patterns of light are carried from the retina to the brain in the optic nerve on the right. The first task of the eye is to adjust the amount of light that gets into it and make sure that it's neither too bright nor too dim. In a situation like this, Going from sunlight to shade can produce variations in intensity of many thousands of times. The amount of light is controlled by the iris, the colored area around the pupil, that black hole in the middle of the eye. Depending on the brightness of the light that falls on the eye, the pupil will get larger or smaller. And this change happens very quickly. The iris is able to respond immediately to the moment-by-moment changes in brightness that happen at a time like this. These rapid variations in size are due to the action of two sets of tiny muscles. Here we're looking at them from inside the eye. One set of muscles radiates outwards from the center. When they contract, they make the pupil bigger. This happens in dim light. There are also circular muscles and rings around the pupil. When they contract, the pupil gets smaller. This happens in bright light. As well as responding to light, these muscles appear to react to emotional images. The pupil dilates when we see something attractive and constricts to unpleasant images. Behind the iris is the lens. We've made it red here to make it more visible, although it's really transparent. By a simple change of shape, it can bring an image into focus on the retina, the projection screen for pictures of our world. As the lens changes shape, it bends the light rays from the objects we're looking at and makes sure that they meet exactly at the back of the eye. These changes in the iris and the lens happen automatically. When this information from both eyes is fed back to the brain, we can use it to tell us about the three-dimensional qualities of the outside world. For example, the changes in lens shape tell us how near or far something is. If the driver focuses on the dangling monkey, his lens is much more curved than when he focuses on the road ahead. So all the information from our complex visual world travels through the pupil and the lens of the eye to arrive eventually on the retina. In this remarkable view of a living human retina, we can actually see a reflection of the pupil seen from the other side. It's that dark spot in the middle of the screen. The retina is only the size of a postage stamp, and yet it has to receive and pass on all the color, brightness, and sharpness of everything we can see. One surprising feature of the retina is that its surface is covered with blood vessels and nerve cells, and yet somehow the brain is able to ignore the obstructions. Otherwise, we'd see the world like this. The patterns of brightness and color are spread over 125 million light-detecting cells. There are two types called rods and cones. In the center, they're much closer together because that's where we direct the part of the picture that we want to look at most closely. Each rod or cone is a cylindrical cell containing a chemical pigment that becomes bleached every time a light ray hits it. The light, coming from the top of the screen, hits the pigment, which is contained in these layers of discs.
The video just kind of cuts off, but now let's talk more about the rods and cones. The cones are mostly located at this area of the eye. It's called the Fovea Centralis. And it's at the location where if you're looking right at something, it's focused, the light is being focused on the fovea centralis. So, look up here at this word deuterostomes. You're able to read those words because when you're looking right at this, all the light is going into your eye and hitting the fovea centralis where the cones are. Cones can make out detail. So you're able to read deuterostomes. But you probably aren't able to read this word right here if you're looking at deuterostomes. You aren't able to read here. Because the light coming from here is not hitting your cones, it's hitting your rods. And rods can't make out detail. So, rods are good at seeing movement though. So if you look at deuterostomes, you could probably see my hand moving because the rods can make out movement. So rods are located everywhere else on the eye. Cones are mostly at the fovea centralis. There are some cones scattered around elsewhere, but most of them are at the fovea centralis. So, keeping that in mind, let's take a look at how these cells are wired. These pink ones are cones. If you notice, so light actually comes in this way, and the light passes through all these cells, which are kind of clear in color. The light goes through all these cells and hits a rod or a cone. These are cones, these are rods. Now if it hits a cone, the cone has its own personal connection with the brain. You can see this cone will fire off a signal, pass it to this neuron, and this neuron will pass it to this neuron, and that neuron goes to the brain. So every cone has a direct signal to the brain. Look how the rods are wired. Here we have 3 rods. If light hits there, or there, or there, they synapse here with these, and that synapses with this, and this goes to the brain. So the brain would know if this cone was sending a signal, but the brain wouldn't know if this cone, if this rod or this rod were sending a signal. The brain just knows it's one of those 3. It doesn't know which one it is because they all converge onto the same neuron that goes to the brain. So that's why these rods can't make out detail. The brain is not exactly sure where the light is coming from. It just knows it's from a general area. So the rods just see in generalities. Again, if you're looking here, this writing is hitting your rods, and the rods can't read it. The rods can only make out generalities. Wouldn't it be cool if your entire retina was cones? Then you wouldn't have to look right at something to read it. You could make out the detail not looking at it. But the cones are very energy intensive. Each cone, the cones come in 3 different colors. There's a red cone, a blue cone, and a green cone. And the combination of those give you the color. And the cones end up, because there's so many different types, they end up using a lot more energy. So you save energy if you have rods. It's energy expensive to have cones. Each cone requires its own attachment. And so what evolution has done is it's put cones in areas where when we're looking, we're only in one spot, so we can make out detail. And it put in the muscles of the eyes to turn the eye toward the thing that you're interested in. So the rods can detect some movement. So if something's moving over here, then you look right at it. Then you figure out what it is with the detailed cones. And that saves a lot more energy than having your entire eye full of cones. So it's kind of interesting how the body has worked to save energy. There are animals with only rods. Those, like a deer, can't see color. That's because its iris has all rods. It doesn't have any cones. So a deer would look at this word and it wouldn't see it couldn't read deuterostomes Even if you tried to teach it, it can't pick up that detail. It would just see kind of a fuzzy gray area. So deer can't see color, so because they don't have cones, you see, so you can wear an orange vest if you're hunting deer. I think that's cool. Dogs the same way. Dogs can't really see color. Now some of these animals have some cones, but not many. Birds have eyes just like ours, basically. And so you got to wear camouflage if you're hunting birds, because they'll see an orange vest. Deer can make out movement real well, though, because they have the rods. So you got to be still when you're hunting the deer.
You a hunter?
I used to hunt when I was a kid. I never could shoot a deer, though. I just felt too bad for the deer. I had a shot one time, and my dad was with me. He's going, shoot, shoot. I'm like, I can't. Plus, I'm not a very good shot. Doesn't help. I like shooting skeet. Here's another vision video.
To form images of our environment, light emitted by or reflected from objects must be focused on the retina.
Watch it.
Some of the light reflected from the surface of a yield sign is directed toward the eye. Upon reaching the eye, the light is first refracted by the cornea to send it on a path toward the retina. To reach the retina, the light rays must pass through the lens, where a small amount of refraction adjusts the angle of the light to form a focused image on the retina. Central to the sense of vision is the transduction of light energy into electrical energy by cells in the retina called rods and cones. Cylindrical-shaped cells called rods are responsible for the perception of light and dark. Conically shaped cells called cones are sensitive to red, green, or blue light.
That shows real well. 3 cones to 1 signal going to the brain. I'm sorry, 3 rods to 1 signal going to the brain. 1 cone to 1 signal going to the brain. It's interesting that to see something, the light has to come in, it has to hit the rod, then the rod has to send a nerve signal that goes through these several neurons, through synapses, and that goes all the way to the brain and goes to which lobe of the brain? Do you remember is responsible for sight?
Occipital.
The occipital lobe in the back of the brain, and only then do you actually see something. But doesn't it feel like you're seeing it right when it happens? It's quick. It's amazing how fast the system works. You also have a blind spot. You know about your blind spot? There's a point, to go back to this picture here. There's a point where all the blood vessels come in to spread over the surface of the retina. And that point there does not have any visual receptors. So you have what's called a blind spot. And it's not hard to find your blind spot. Do me a favor, if you have a pen and a piece of paper, put a dot right there, somewhere on your paper, and a plus right there, about that far away from it. And then what you want to do, so a dot on the left and a plus on the right, about 1/3 of a page apart. And then what you do is you cover your left eye and you stare at the dot with your right eye, with your open eye. And then just move your head toward the book. And at some point, if you're staring at the dot, the plus will disappear. That proves you have a blind spot. Can you find it, Queen Kathleen?
I found it.
How far away from the book is it for you?
Right here.
Yours is farther away from the book. I think the older you get, the closer it gets. The blind spot gets to the book for some reason.
I do have to do.
It's interesting that you don't notice that blind spot when you're looking at things because you have 2 eyes. People with only 1 eye. I have a buddy that I play he only has one working eye. He's got two eyes, but only one works. And you wouldn't know it because he's probably the best golfer in the county. His name's Josh Williams. But he says when he's lining up a putt and he's looking with that one eye at the hole, his ball will disappear. And that because it's in his blind spot. But with two eyes, because the blind spot is in a different location in each eye, you don't notice your blind spot. Isn't that interesting? And by the way, what does your eye color the page where the plus sign was? Colors it white. Your eye fills in missing information with what it thinks ought to be there. Your brain fills in missing information with what it thinks ought to be there. Hey, we couldn't do IDs today because the other class was in there.